Welcome viewers. Today in this session we will talk about RNA. What is RNA? What are the different functions which RNA plays inside a living cell and how RNA was replaced by DNA in course of evolution and how the different functions of RNA or structures of RNA was studied by the eminent scientists through time. Let us move to the RNA. So what is RNA? RNA stands for ribonucleic acid and is one of the most important nucleic acids found in a living cell. And the functions of RNA are more profound and diverse than DNA. Besides, RNA also serves as a genetic material in some organisms known as RNA viruses. The functions of RNA. So what are the functions it plays? The first function is it is known to play an important role in protein synthesis. We know that the different types of RNA like mRNA, tRNA, they play an important role in protein synthesis. The second is RNA plays an important structural role like we have the rRNA in ribosomes. So these ribosomal RNA play an important structural component of the ribosomes found both in prokaryotes and eukaryotic system. The third function is RNA, it has been shown to contain a catalytic activity. Like proteins, it can form certain or it can catalyze certain enzymatic reaction in the form of ribozymes. And the fourth but not the last is the regulatory function. It plays an important role in modulating the gene expression of living cells as seen in the case of small nuclear RNAs, siRNA, miRNA and riboswitches. So after knowing about all these functions of RNA, we should be moving to the RNA world. So seeing the diverse function of RNA which it plays inside a living cell, it was put forward that RNA serves as the progenitor molecule for giving rise to origin of life. Because it functions as an enzymatic reaction, it is functioning as a structural component in regulating the gene expression, so it was thought so. The information giving rise to origin of life on this earth is encoded in the 4-letter alphabets. Besides, it can take up the various structural forms in greater number than DNA and it can reproduce by itself. So all these processes or all these properties might have helped RNA to give rise to origin of life on this planet Earth. Now let us come to the section where experimentally it was proved that RNA serves as genetic material in living organism. The experiment of such a kind was first performed by Conrad and Singer in the year of 1956. And what did they saw? They used the model system, that is the tobacco mosaic virus. Tobacco mosaic virus is a virus which is known to cause disease in tobacco. Now what did they do? They used two different types of tobacco mosaic virus. One is type A and type B. Then they degraded it with the help of enzymes. So they were able to isolate the RNA and the protein component of the two different types from the tobacco mosaic virus. They produced hybrid TMV viruses. They mixed the RNA from type A and the protein from type B to produce hybrid viruses. After the formation of hybrid TMV, they used the hybrid TMV for infection of tobacco leaves. After the infection of tobacco leaves, it produced progeny viruses. On further examination or on close examination of progeny viruses, they found that it is the RNA component present in the hybrid tobacco mosaic virus, which determines the type of RNA and protein present in the progeny virus. So, in this way, they were able to prove experimentally that it is the RNA and not the protein which serves as genetic material in these kind of viruses.
Ma'am, since RNA is found as the genome of several viruses, why was it at all replaced by DNA?
Now, we can see after the diverse nature of RNA that RNA came before to DNA. Now, why RNA was replaced by DNA? Let us come to the chemical structure of RNA. So, RNA, it is made up of ribose sugar, whereas DNA consists of 2-deoxy-D-ribose sugar. When a nucleophile such as an hydroxyl radical is present in the cell, it attacks the carbon 2' position of ribose sugar, thereby abstracting the hydrogen ion from the hydroxyl group. It is left with the negative O- ion, which interacts or it charges or attacks the delta P plus or delta plus P of the phosphodiester backbone of RNA chain. In this way, as you can see in this diagram, hydroxyl radical is attacking it. As a result, what happens? A cyclic intermediate is formed between the carbon 2' position and 3' position of the ribose sugar present in RNA. This intermediate is known as cyclic 2', 3' intermediate. Now, in the presence of water, that it undergoes hydrolytic cleavage. The phosphodiester bond then is broken down by water. As a result, the sugar backbone is cleaved and RNA chain is shortened. And what are the products formed? Now, depending if the phosphate group is retained on the carbon 2' position, then the product is known as 2' phosphate product. And if the phosphate group is retained on the carbon 3' position, it is known as the 3' phosphate product. So, what we can say, it is precisely attacking the 2' carbon atom of the ribose sugar. In case of DNA, oxygen is absent in the carbon 2' position, and hence the nucleophile like hydroxyl radical is unable to cleave the phosphodiester backbone of DNA. So, DNA thereby is more stable than RNA, and therefore, in course of evolution, RNA was replaced by DNA to serve as the genetic material in the living organisms. We know now more about RNA. This is due to the eminent researchers or research groups working in different fields revealing the different functions of RNA. So, it's not possible to pay tribute or homage to all of them, but we can mention a few names. Like the first of such a kind was started by Severo Ochoa, for which he was awarded the Nobel Prize in 1959. He isolated an enzyme which can synthesize RNA in the laboratory conditions. Similarly, we know Robert Holle. Robert Holle is famous for the structural elucidation of alanine tRNA isolated from yeast. He was awarded Nobel Prize along with Marshall Nirenberg and Hargobind Khurana. So, what did they do? Hargobind Khurana, he was an Indian biochemist, and he was able to identify or show how the genetic material present in the living cell can be converted to proteins. So, he helped in the elucidation or in deciphering the genetic code. Similarly, we have Marshall Warren Nirenberg. He played an important role in describing how the genetic code operates and how it helps in protein synthesis. Further on, we have David Baltimore. David Baltimore, along with the other researchers, were able to isolate the enzyme reverse transcriptase. We know now reverse transcriptase plays an important role in the pathogenesis of infections caused by HIV. Similarly, we have other works that is done by Thomas Cech. Thomas Cech in the year of 1980s was able to show the catalytic activity of RNA in the form of ribozymes. He isolated it from tetrahymena and prove or show how it can catalyze the activity or enzymatic reactions. Further on, we have Andrew Fire and Craig Mello, who showed how RNA can play a regulatory role in the developmental processes. They used the system C. elegans and they proved that it contained siRNA or microRNAs which played a significant role in the development of C. elegans. For significant work, they was awarded Nobel Prize in 2006. So still experiments are being carried on in the field of RNA to prove or to show the diverse functions of RNA found in us. What or how the structure of RNA looks like. If we look at the structure of RNA, it is made up of ribonucleotides. Ribonucleotide in turn is made up of three components. One is a nucleobase, second is a ribose sugar, and third is a phosphate. There are four main types of bases found in RNA. They are adenine, guanine, cytosine, and uracil. Now these three units, that is ribose, nucleobase, and the phosphate group makes a ribonucleotide. How? If we look at the diagram, we can see that this is the ribose sugar. The carbon 1' position of the ribose sugar is held on to the nucleobase, which it can be anything out of the four bases. The base is held to the ribose sugar by the means of a glycosidic bond, which is a covalent bond. Similarly, we have the phosphate group held to the ribose sugar at the carbon 5' position. So this whole together is one ribonucleotide. So RNA is made up of ribonucleotides. Now besides the four bases which we find in RNA, RNA is also known to contain modified nucleosides. The examples of which are inosine, dihydrouridine, pseudouridine, ribothymidine, etc. What are the functions of modified nucleosides in a living cell or in a RNA? The first function is it is now known to confer increased stability to the RNA molecule in which it occurs, like increased stability to tRNAs, because we know that tRNA contain pseudouridine dihydrouridine. So it produces stability to it. Moreover, it helps in the recognition by proteins and other RNAs in the translational apparatus. So it helps in the folding also. The third is some enzymes like amino acid tRNA synthetases recognizes the modified bases, and thus they are able to add on to the amino acids to the tRNA. So in this way we can see that how modified nucleosides play an important function in the RNA. Now let us move to the base pairs found in RNA. We know that there are mainly two types of base pairs. One is AT and GC, that is adenine always base pairs with thymine and guanine always base pairs with cytosine. But in case of RNA, we find another unique base pair, that is guanine base pairing with uracil. So this is known as GU base pair, found only in RNA. The stability of base pair, if we look at, is greater in GC, followed by AU and then by GU. Because of the presence of an additional base pairing, the RNA has an increased tendency to form secondary structure within itself. Now let us move to the different structures of RNA, or the structures of RNA which it can adapt. So first let us move to the primary structure. Now after knowing about ribonucleotides, now we can say what is the primary structure? The primary structure of RNA is a polymer of ribonucleotides. So the ribonucleotides are held on to each other by means of phosphodiester bond. Apart from this, from the primary structure of RNA, we can know the sequence of bases in which it occurs. So, the primary structure of RNA helps us to know about the sequences of bases in which it occurs. Like in this diagram, we can see that this is a RNA chain and it represents the primary structure of RNA. The RNA chain has a polarity, like we have a 5' phosphate group and a 3' hydroxyl group. Each ribonucleotide is held on to each other by phosphodiester bond. Phosphodiester bonds are covalent bond. Accordingly, we can see that the position of the bases or the sequence of bases is fixed. In this diagram, we can see that from 5' end, the sequence is UGAC, while from 3' end, it is CAGU. So, we know the sequence in which it occurs. Let us now move to the secondary structure. In nature, RNA doesn't stay very long in the primary structure. It folds by itself or onto itself to form the secondary structure. Now, these secondary structures are formed between regions of complementary bases followed by regions of non-complementarity. Accordingly, the RNA can take up different secondary structures. What are the different secondary structures it can take up? The first is bulge loop. We can see a bulge is formed on one side of the RNA chain. This bulge is formed due to the region of non-complementarity within itself. Again, we have the internal loop where we are able to see bulges on the two sides, followed by multi-branch junction and stem loop structure. Stem loop structure, also known as hairpins, are the most common secondary structures found in RNA. The first example of stem loop found is tRNA. tRNA contains more than one stem loop. Now, these structures, as I have already mentioned, are formed because of complementary regions followed by non-complementarity regions. So, these are the different secondary structures. Now, what are the forces which are stabilizing the secondary structures? Forces which act on DNA, like the hydrophilic forces or the ionic interactions which is taking place, the covalent bonds, hydrogen bonds, stacking interactions, van der Waals forces also contribute to the stability of the secondary structure of RNA. Apart from the mentioned or the conventional secondary structures of RNA, RNA can also take up the structures called tetraloops and pseudoknots. Tetraloops are found when the bases they stack upon each other. The most common tetraloop structure is formed by sequence UUCG. And here, one of the stabilizing factors for the formation of tetraloop is the base stacking. Base stacking here contributes a significant amount of energy for the formation of tetraloops. Another structure is that of pseudoknots. Pseudoknots are formed by base pairing between regions of non-continuous in nature. So, the base pairing is formed because of pairing potential, but the regions are not close to each other. So this is again leading to the formation of a complex kind of structure. So now let us see what kind of conformation does the secondary structure takes up in case of RNA. We know in DNA the secondary structure is generally the B form, while in case of RNA it takes up the A form of conformation. The A form of conformation of RNA is taken up because of the fact that RNA contains ribose sugar. Because of the presence of C2' hydroxyl group, RNA generally takes up the A form of helix. As a result, the RNA binding proteins are able to bind only through the minor groove. And we know that minor groove is less rich in chemical information than major groove. As a result, proteins binding to RNA are few in number in comparison to DNA binding proteins. This is the picture where we can see the difference between the A and the B form. Here the minor groups is wide and they are narrow. As a result, it is not able to accommodate many of the proteins. Whereas in case of DNA, most of the proteins bind via the wide major groove, which is able to accommodate proteins as well as it is rich in chemical information.
Ma'am, this tertiary structure of RNA, does it in any way help in the catalytic part of RNA?
The tertiary structure is formed because to perform the catalytic function of RNA. Let us now move to the tertiary structure. The tertiary structure is formed when the secondary structure folds onto itself in a three-dimensional way. So this gives rise to the tertiary structure. Now, it is due to the why in RNA the number of tertiary structure is greater. It is because of the rotational freedom which it has in the non-complementarity based regions. So, the sugar is or the sugar backbone is able to rotate. Moreover, it is due to the formation of base triples like UAU and the other structures which contributes to the formation of tertiary structure of RNA. Here, the bases which are present in the non-complementarity regions interacts with the sugar phosphate backbone present in other part of RNA in the same molecule. As a result, the tertiary structure is formed. Now, the tertiary structure is performing important functions like we see it in tRNA. tRNA, the amino acid acceptor site, is exposed when the tRNA is folded into a tertiary structure. Accordingly, the anticodon loop is also made available for binding to the mRNA. As a result, it is able to function more efficiently when the tRNA takes up the tertiary structure. Now let us move to the differences with which it has with DNA. The differences can be divided into two main categories. One is the chemical difference and the structural difference. The chemical differences, in case of DNA, we have 2-deoxy-D-ribose, while in case of RNA, it is ribose sugar. Now we know that since it contains ribose sugar, it is more susceptible to cleavage by nucleophilic agent, whereas in DNA it is not possible owing to the absence of oxygen atom at carbon 2' position of the sugar. Moreover, DNA contains thymine, or thymine is specifically found in DNA, whereas RNA contains uracil. Now the question comes in, why RNA contains uracil? Because cytosine can deaminate in vivo spontaneously to form uracil. Had DNA contained uracil, it would not been able to distinguish between uracil generated from cytosine. As a result, mutation would have occurred. So, in order to prevent or distinguish between uracil generated from cytosine and the uracil present as such on the DNA molecule, the thymine is produced. So, what is thymine? Thymine is the methylated derivative of uracil. Now, let us come to the difference in terms of structure. Structurally, RNA is shorter than DNA in terms of nucleotides, and therefore, the number of structures or secondary and tertiary structures which RNA can take up is far greater than DNA. Along with the secondary structure of RNA takes up the A conformation, while the secondary structure of DNA takes up the B conformation. After knowing about all these structures and important functions of RNA and how RNA was discovered, slowly with time, we now come to the conclusion. So, what we can conclude from after knowing about all this? Now we know that RNA is one of the most important molecules. RNA could serve or serves as genetic material in some viruses known as RNA viruses. RNA performs more diverse functions than DNA. The first function in terms of protein synthesis. Second, it plays a structural component in organelles like ribosomes. It can perform certain catalytic activities similarly to enzymes. It can modulate or change the gene expression as seen in case of siRNA, miRNA, etc. or riboswitches found in the prokaryotic system. So, after knowing all these functions of RNA, we can say that RNA came before DNA. So, accordingly, it was put forward that RNA gave rise to origin of life on this earth. This hypothesis, known as RNA world hypothesis, was put forward. Moreover, due to the significant contributions made by different researchers in different fields of RNA, we were able to know more about RNA. So, in this way, we can say that RNA plays a multifaceted role in living cells.